Hello, everyone, and welcome to today's webinar, Characterization of RNA Therapeutics from Small to Large. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by cIEF. To learn more, visit cIEF.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Steve Locke, Ph.D., Biopharma European Marketing and Market Development Manager. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Steve, you may now begin your presentation.
Thank you for that great introduction. Today I'll be talking about, as mentioned, the characterization of RNA therapies from small to large. And I've been working now in the biopharma industry for nearly over 20 years, supporting the industry with developing methods for characterizing biopharmaceuticals. And RNA is one of the growing areas of research in the biopharmaceutical industry. So what I'd like today is to go through some of the solutions that science are producing for this space. The talk itself is outlined on this slide, so I'll go through a bit about the background for RNA therapy, and then I'll look at the techniques used to characterize small oligonucleotides and move on to larger oligonucleotides, which are really RNA molecules. And then I will finish with some conclusions from some of the aspects I will discuss today. So, as mentioned in my outline, now I'd like to move to the background to RNA therapies. And this slide, I think probably already a lot of people on the call already know this, gives sort of a highlight of where RNA fits and why companies are targeting RNA as a therapeutic area. The conventional view of RNA is that it transfers information, but to be honest, it also acts in several other ways within the body, and it helps to turn genes on and off. It aids chemical reactions. It also alters other RNA molecules and helps to build proteins, and this is via transcription and reverse transcription. So it's actually pivotal in the information distribution within the body and within how a cell works. So by altering RNA, you can have a big impact in how a cell works and a big impact on disease. So looking at some of the actions of the RNA therapies themselves, and they can sort of be sorted out into three broad categories. The first one of these is that RNA therapies which target nucleic acids, either DNA and RNA. There's RNA therapies that target proteins and those that encode to proteins, so they actually have the information to generate proteins within cells. And there's also hybrid approaches that combine several RNA-based mechanisms into a single package. And these are currently the more recent flavor of RNA therapies, and they're starting to emerge now in biopharmaceutical research. So looking at the RNA therapies in more depth, I suppose one of the ones which is released and actually is FDA-approved drugs these days is Antisense Oligonucleotides, or ASOs. And these sort of small groups of bases, or small stringer bases, prevent our mRNA from working and building proteins, or alter splicing or stopping a precursor mRNA from becoming active. They're typically small. So, for example, they range from 13 to 25 nucleotides. And because of their size, they actually can be assayed using techniques like LC-MS, and I'll discuss that technique in a minute. You also have siRNA, or small interfering RNA, and these help to degrade a messenger RNA, preventing it translating into target proteins. And again, these can also be small, but tend to be double-stranded in nature, unlike the single-stranded nucleotides you get from ASOs. You also have RNA aptamers, and these bind to specific targets like proteins, and can help also deliver siRNA into cells. And they're generally a lot bigger than siRNA at 50 to 120 base pairs, nucleotides long. And then finally have messenger RNA itself, and these are used in vaccines. And I think a hot topic is messenger RNA is the use of biopharmaceutical companies of this type of RNA in the development of COVID vaccines, for example. And these tend to be thousands of bases in length. So we're talking of greater often than 10,000 nucleotides in length. So as you can see, the sort of RNA therapies range from small ones, 13 to 25 single-stranded nucleotides, double-stranded nucleotides, up to very large nucleotide lengths of over 1,000, 10,000 even, nucleotides in length. So now I want to move back to, I suppose, the first area of this, which is ASOs, or small oligonucleotides. And this slide gives a highlight of the synthesis and metabolism of small oligonucleotides. Now, I suppose the synthesis of oligonucleotide is like a building block, like sticking Lego bricks together. You tend to add bases on base, and these can be synthesized and are not necessarily built in cells. Okay, these are synthesized, and these are generally 3 to 5 prime serial synthesis using phosphoramide building blocks. Okay, and the chart given here on the left-hand side represents the sort of the model impurity plot based on the synthesis. Now, you can assume that as the ASO or the oligonucleotide gets longer, then basically the probability of increased impurities also increases. So generally, every time you add a bit on to the oligonucleotide chain, you also increase the impurities present. So it tends to have diverse and mixed populations of impurities when you generate these oligonucleotides via synthesis. Okay. With regards to metabolism, you can see that this ASO or siRNA are typically clipped by the 5 and 3 prime, and they're clipped in vitro and by exonucleotases, as shown on the right-hand side of the slide. Therefore, the sort of their intercellular and circulatory levels of M and 3 prime metabolites tend to be mixed. Okay, they also involved in ligand binding assays can also over represent the metabolites here. So you've got to be careful using some assays, and this is why people are looking at LCMS based approaches, which are more specific than ligand binding assays. So again, looking at some of the shortcomings of existing oligonucleotide assays, and here you can see them highlighted in these sort of red boxes highlights where some of these shortcomings are. I'm talking a sort of ELISA or ligand binding assay based formats of testing. And I think if you look through, you see inadequate sensitivity is one thing, less so with ligand binding assay, of course. Insufficient selectivity, and this is a problem sometimes specifically with ELISA or sometimes there'll be assays, and also too much rigidity. Okay, so many assays are fit for purpose, exactly compliant assays. They're not flexible enough to be used in development. Okay, so you want something which can be essentially flexible to be used in development, but also be pushed into QA/QC. And often when problems happen in manufacturing because the assay is so rigidly based on one structure, and perhaps another structure is formed, it's difficult to change the assay to respond to a problem which is arised in manufacturing. People again looking at flexible techniques which are adaptable to some of the challenges you can see in manufacturing sometimes. I suppose this brings into the use of LCMS in helping to answer some of these analytical challenges of oligonucleotides, so ASO therapy production. Now, there's some of the characteristics of an LCMS assay which are needed for oligonucleotides are listed on this particular slide. I think one of the things that's most important to point out here, it's not like looking at proteins which are positively charged and tend to be easy to detect. Oligonucleotides are in negative ion mode because they're essentially acidic in nature, so that can have some effect on its sensitivity. The other thing which is really important to bear in mind here that often the chromatography is complicated because you have to use ion pairing reagents. So there's a balance between on-column retention of your analyte or your oligonucleotide and the suppression you get from that ion pairing reagent. And there's a problem with using ion pairing reagents in that they can potentially contaminate your mass spectrometer as well. So you've got to be careful of the amounts of ion pairing reagents used. And, I've actually encountered ion pairing contamination in LC systems before now. And typically people working and using lots of high concentration ion pairing reagents tend to have a dedicated LC system for this, or they have to do extensive cleaning afterwards. So ion pairing reagents are essentially a pain to handle sometimes, and their impact on swapping between analysis on the same system. The other thing you have to bear in mind is that these type of analytes quite easily form adducts, particularly with alkali metals such as sodium. So sodium salts presence can produce adduct formation, which will complex the fragmentation of these molecules if you want to do MS/MS analysis. And typically you also have wide charge state distribution as well, which can affect the sensitivity sometimes. And with the wide charge state distribution, fragmentation efficiency, so how well the molecule breaks up in the gas cell or mass spectrometer, can be dependent on the charge state. And you typically have to select a more highly charged, higher charge state ion to get a better fragmentation profile. Finally, sample preparation recovery. You've got to be careful here. So you need strong oligonucleotide protein binding in the matrix can happen. So you've got to be careful of that as well when you're trying to release the oligonucleotides for the analysis. So looking at the sort of technology that science promotes for this space, well, what we try to promote is a mixture of mass spectrometry depending on exactly what you want to do. If you want to characterize on an intact level, we probably recommend using a QTOF system, for example, the 6600 triple TOF or the X500B QTOF mass spectrometer. We tend to recommend the use of microflow. Microflow increases the sensitivity because you improve ionization efficiency, but also decreases the use of ion pairing reagents and their flow into the mass spectrometer, so improves the robustness of the overall assay. So this is typically what we recommend when we do oligonucleotide analysis, and we partner this all together with software, for example, cIEF, and working with third-party software like ProMass to help with the data manipulation that you require to do the analysis. And here you see some examples, and you can see that the 5 and 3' capped oligonucleotides here, and this is the sort of multiple charge envelope shown on an accurate mass system. So this would be a QTOF system from CIEF, and this allows you to confirm the mass of the target, because this shows you that this oligonucleotide has a molecular weight of just under 7,000 AMUs. And also helps you with confirming its sequence, because the molecular weight itself gives you an indication of the sequence of the oligonucleotide. And we use detection at the intact level to help us with failure sequence identification. And here we see the high quality of spectra due to source and ion guide technology. So what really helps here is the improvements in quality of spectra due to the design of the mass spectrometer interface. So the use of the ion drive and the use of the QJet enables really intense major parent ion and less presence of things like sodium adducts, which can interfere with the spectrum. What you're seeing here is in the middle of this page is this sort of the master charge envelope. So you see the -9 charge state down to the -3 charge state, and inset is the deconvoluted intact molecular weight of the oligonucleotide compared to the overall spectra you'll get. So this mass spec you can see, and the quality of the mass spectrum you get on a QTOF system science helps you identify the target sequences and helps you identify failure sequences in your oligonucleotide preparation. And here you can see some examples of how the Q1 spectra changes depending on the sequence of the oligonucleotide you synthesized. And then moving on to the next slide, then what you can also do then is help to identify and confirm the sequence by actually doing MS/MS fragmentation. In this case, as mentioned, you would pick the most highly charged parentine in the spectrum, in this case the -9 charge state, and then fragment that in the gas cell. And from the spectra you get, you can sequence the oligonucleotide present, and this helps confirm the sequence. And again, because you can do this quite efficiently on a QTOF system, you can also then help confirm the sequence by looking at some of the secondary ions as well. And here we see just an example of the secondary ions detected and the confirmation you can get when you're running this type of assay on the QTOF system. So looking at failure sequence, one of the best ways to do this is to automatically process the data. And this can be done using software like ProMass. And this example here shows you what you can get from the data output of ProMass. So we would do an LC-MS analysis using a SELEX system, and then we would take the data files you get from that and then process them through ProMass. And that then helps by generating an HTML summary sample report, which you can pick up the failure sequences present in the sample you run. Okay, and here just shows you some of the purities obtained from these different the peaks, and it shows you the confirmation of the data you get from ProMass data analysis of data acquired on a SELEX mass spectrometer. Again, this table shows you a failure sequence report and a purity report from ProMass, and shows you where the failure sequences were present from a 21 mer ASO oligonucleotide. Okay. Again, this slide's quite small, but it gives you an idea of the sample export data you'll get from ProMass. Okay. And again, deep coverage of the failure sequence can be done by going back into the MS/MS spectra and looking to confirm the presence of specific fragment ion peaks in the data. And this just shows you presence of some of the failure sequences in the raw data itself. So if you look back, you can see N-4, N-4 fine prime peaks which have been detected in the LC-MS analysis. Okay, so LC-MS typically is used, I would say, for your smaller RNA molecules, so like your ASOs analysis. When you come to larger molecules, you typically move to a different technique. In this case, you move now to capillary electrophoresis to help profile the impurities present in these larger molecules. So in the next couple of slides, in the next 10 to 15 minutes, I'll discuss how CE or capillary electrophoresis is used in the characterization of small to large RNA molecules. And even, looking at some RNA molecules which are greater than a couple of thousand bases long. So basically CE offers a convenient method for exact size determination of an RNA molecule, and this comes from using automated capillary gel electrophoresis. So basically, what we're doing here is we're offering a gel electrophoresis separation within a capillary. And at SELEX, we provide gels to capillaries to do this. And this is what's listed on this side of the SELEX ssDNA 100R kit, which is available for the PA100 Plus system from SELEX. And this allows you to profile small oligonucleotides and to show you separate them by a single base separation from 10 to 100 bases long. The single base resolution is important because it allows you to pick up failure sequences present. And on the inset, you'll see here an example electropherogram where we're separating a mixture of RNA molecules from 4 to 6 bases in length. This basically, every time you run a sample, you completely replace the gel between injections, so there's no carryover in this system. And to be honest, we don't use things like ion pairing agents. It's a gel electrophoresis-based separation using gel specifically designed for RNA. It's a good method to process and look at purity during development. Two examples of this where it's been applied to siRNA therapies for purity is shown in this slide here. Again, siRNA and ssDNA, single-stranded DNA, are quite small in size, and these ones, two examples here, are 10 base pairs long, the siRNA, and 45 base pairs long. And what you'll see is the major peak here is the major compound, the major type of RNA molecule present. And the minor peaks tend to be specifically on the 45 base ssDNA. These are basically 44mers and basically show a single base clipping from the parent 45 base ssDNA. And here's an example where this has been used in process analytical technology and helped in process development. In this case, this is a small 25-mer tritillated phosphorothioate oligonucleotide, and this actually application is done. An application note is present on the Sativa's website. This was originally done by Amersham Biosciences, and we can show here how CE is being used to look at the final product and compare it with the process crude sample, and to show if the non-tritillated failure sequences have been separated away from the product pool. And the three panes here show the crude sample in the middle, on the far left-hand side, and we see the middle non-tritillated failure sequences which have been separated away from the final product pool. And this shows you how CE can aid process development in oligonucleotide manufacturing. The next slide is really around RNA aptamer manufacturing, and it's a quite clever way that CE can be used to help, again, select and help to produce aptamers, RNA aptamer therapies. And in here, we're actually using CE as a fractionation device. So basically, we got CE to separate the aptamers, right? And then you collect the fractions, and then you use use PCR to multiply the fractions, and then repeat the process. This process is called CE SELEX, and basically, so what you're basically doing is using CE as a fraction collection and a separation device. And it's used in a workflow for aptamer production, and there's a group at Oxford University have used this type of workflow. And here you can see some of the key advantages. And there's an application note for anyone interested in using CE for aptamer RNA production, which is present on the CIEF website. And here you can see some of the key advantages of CE SELEX. It is basically what it does, because it automates the process, but it also means you actually need fewer rounds of selection using this approach, which again speeds up the selection of the aptamer RNA, which you want to purify. There's less, because you're using CE, and it's really high resolving technique for separating off different forms of the aptamers. Is less opportunity for non-specific interactions. So, and the yields many independent sequences. Again, that's due to the resolution, right? So it allows you, because you're fraction collecting, to capture different sequences. So it's quite a flexible technique for aptamer RNA production. And anyone interested in this particular area, I recommend to go to the CIEF website and download the application note for this particular space. But the other thing really is really around as you move to larger molecules of RNA, as I mentioned, and CE is capable by changing the gels and the capillaries you use to separate even larger molecules in RNA, and it's very, very reproducible technique. CE and gel-based separations have been used in the biopharmaceutical industry for over 15 years for analysis of proteins, for example, in quality control of monoclonal antibodies. And actually, CE separation of DNA and RNA has been around for a similar amount of time. So it's a well-recognized technique, and it actually can really help with the separation of size-based impurities in manufacturing of messenger RNA and smaller RNA. And on this slide, we show some examples of the resolution you get. And this is using commercial ladders. So these are ladders you can buy from companies like Thermo Fisher. And you can see that on the far left-hand side of the first example, you'll see the separation of a ladder, a commercial ladder, which is bought in, running from 0.2 kilobases up to, I think it's 6 kilobase size of RNA. And this is highly reproducible. So if you look on the right-hand side here, you can see where we've actually repeated this assay several times, and they clearly overlay one another. So unlike slab gel electrophoresis, this CE is very reproducible and allows you to integrate the peaks, reproducible, and give you accurate percentage determination of impurities present in your sample. Now, often in RNA analysis, you would tend to stick to UV detection, except where you have low concentrations of samples when you move to LIF. And if you look at sort of standard sensitivity here, you'll see that the LOQs you get for in this particular case go down to 0.33 nanograms per mil with an LOD of 0.08 nanograms per mil. So this means you can actually detect very low percentage impurities in your product, and the response is linear over a couple of orders of magnitude, as shown on the right-hand side of the slide. And again, this is available from an application, as you can see, which was written a couple of years ago in 2018. So if you want more information on sort of the sensitivity possible for RNA quantification, the linearity, it's available again on the SciEX application note. But a lot of companies are now are looking at using RNA specifically with laser-induced fluorescence to get down to the detection levels required. Typically, when you use laser-induced fluorescence detection in CE, you derivatize the genetic material in order to pick it up. So if you're using single-stranded or like RNA molecules, you use dyes like cyber green to label the RNA, and if you're using double-stranded, you would use intercalating dyes like the DNA molecule to label up the molecule for detection by laser-induced fluorescence. But to be honest, it's not just biopharma manufacturers working in RNA, but it's also the gene therapy industry, as well as using CE now for RNA purity determination. The standard kit we do and have a method for this now is again, as I mentioned before, the P100 Plus system. And for LIF, we use an excitation voltage of 488 nanometers and an emission of 320 nanometers. And we've made this easy to implement because we use a pre-built cartridge, which means you no longer have to burn your windows and cut your cartridge and fit your cartridge into your cartridge, your capillary into your cartridge holder. It's all pre-built now. So most methods which are coming out from SciEX these days use this pre-built cartridge to aid method installation within customers' labs. So how is CE being used in different types of RNA therapy research? Well, it's been used for a variety of different sizes of RNA, and this just shows you an example of CE being used in the CRISPR-based products. And here you tend to combine two molecules for the CRISPR therapy, and you have a single guide RNA and a Cas9 messenger RNA. And here you can run both the same molecules, which are combined for CRISPR, on the CE system, and the CE is capable of then detecting these molecules and identifying the percentage impurities present. You can see here that for the Cas9 messenger RNA, the impurity level seems to be slightly higher because the peaks are higher intensity before the main messenger RNA peak on the electrophoregram on the right-hand side of the slide. So just again, looking at another example on how this compares to a ladder. The Cas mRNA I showed you previously is now being overlaid on a ladder. This is an RNA ladder to give you a sort of a calculation for the size of the RNA molecule. So this is really around about 4 kilobase in size, 4.5 kilobase in size. That's the red trace overlaid on the ladder, which is the blue trace. If you look on the right-hand side of this slide, you'll see another example of the detection of an RNA molecule using this approach. And this RNA molecule is used in sort of a gene therapy medicine. So it's actually in presence within a an AAV viral vector. AAV stands for adeno-associated virus vector. So what we did for this particular assay is that we opened up the viral vector. We basically captured the genetic material, and then we, after capturing and using QA quick kits, which are commercially available, then we just assayed using a similar kit to the Cas mRNA kit. And here you can see the intact genome of the genetic material held within the viral vector, and the partial genome, single small impurities around about 9 minutes, with the main peaks around about 12 minutes. In this case, the intact genome is around about 4 kilobase in size. So again, these are similar sort of sizes of RNA being detected here, but you can go to a lot larger size molecules in RNA. I'm talking over 10,000 bases in length. And typically what you do is you would just adjust your gel that you're using inside the capillary for separation. You can dilute the gel with TBE, and that helps sometimes the analysis of the larger RNA molecules. And again, there's several companies now in Europe which are investigating the use of CE for the detecting process impurities of larger RNA molecules. So I'd like to finish this presentation with a summary. I'll start off with a summary of the oligonucleotide characterization section using LC-MS. And what basically LC-MS offers you is high confidence in confirmation of target sequences, because you not only get accurate mass MS, but also MS-MS confirmation and sequence data for fragmentation patterns. The high quality MS-MS data for sequencing oligonucleotides helps with the confirmation of the sequence. And it also provides well-resolved MS-MS, LC-MS separations of failure sequences, which aid subsequent identification of the impurities present in your product. And CIEFs partner with, ProMass integration and other software to help with the data processing of your samples. If we look at RNA characterization by CE, basically RNA CE characterization helps you profile sRNA and smaller RNA molecules. These typically can be done with UV detection, which enable the process analytical development, but also can be used in LIF, and we tend to move from UV to LIF depending on the sensitivity requirements of the assay. The assay is reproducible and sensitive and provides excellent separation even of larger RNA molecules. And you can simply do this by changing the gel chemistry, and if you want to change sensitivity, as I mentioned, moving to LIF detection. Together with that, on the same Ph.D. plus system, you can do CE SELEX, which helps provide a rapid approach to select and collect aptamers for RNA aptamers for amplification for research. And also, as I mentioned, CE is now being used not only in, in smaller RNA, but larger RNA analysis and in genome integrity of RNA-based products used in gene therapy. At the end of my presentation, I'd like to acknowledge some of the people who've contributed content for this deck and say I don't work in isolation. I work with a great team based in the US as well as the European organization. And this slide just highlights some of the people who've helped me put together this slide deck today. And at the end, I'd just like to thank everyone for their attention today. And now I'm open for questions, and thank you very much.
Thank you, Steve, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Okay, let's get started. Our first question is, can we analyze more than 10 kilobases in RNA purity analysis?
Actually, we can. We've got up to, I think, 12 kilobases or 13 kilobases of RNA. You have to modify your separation gel, those who typically would start to look at diluting the separation gel used to do this. But it is possible to go to the larger RNA lengths as suggested.
Okay, thank you, Steve. Our next question is, what size oligonucleotides have been analyzed by LCMS?
Yeah, good question. I suppose this is really a length-based question. So at CIEF, we've looked at oligonucleotides around about 25 to 30 mer in length. And I suppose what drives this is really the separation of the oligonucleotides on the HPLC column. Okay, so typically very long oligonucleotides are difficult to chromatograph, and you would go down a CE capillary instead. So for LCMS, at the moment, we've looked at sizes up to 25 to 30 mers in length.
And how do the flow rates used in micro LC compare with traditional LC flow rates?
Yeah, again, a good question. I think micro LC typically runs in the range of a couple of microliters up to 10 or 20 microliters flow rates. And with standard analytical LC flow rates, you'll be looking at a couple of hundred microliters. So really you're looking at 50 to 100 folds decrease in flow rates when you go to micro LC. Actually, in one of the application notes you can find on our website, you can actually inject, say, 5 microliters on both systems. And one of the benefits of going to this lower flow rate is the increase in sensitivity you'll get by the improvement in ionization efficiency. And in that particular application note, I think we go from 300 microliters per minute down to 6, which is what we use for the micro separation.
Okay, thank you, Steve. Our next question is, what size RNA molecules have been analyzed by CE?
Yeah, I suppose, again, we've analyzed even the smaller RNA molecules. So, 25, 30 mers can be analyzed. That's the bigger ones, so the bigger messenger RNA molecules. Of course, as I mentioned before, when the listener asked if you could analyze above 10 kilobases, you generally have to adjust your kit. And the larger RNA molecules, you have to typically, dilute your gel. And that's typically what we do. But it ranges from small ones like ASO, or basic oligonucleotides, up to larger messenger RNA. These can all be analyzed with CE on the same system. So all you have to do is modify the chemistries in the capillary, typically.
And can CE separate RNA by shape as well as length?
Yeah, it can. It depends. It depends what you want to do. I think the standard approach for CE is a gel sieving technique. So you would separate simply by sort of the length of the RNA. So you would denature the RNA. So use things like urea in the buffer to denature the RNA. If you take the denaturants out of the separation buffer, then the RNA will fold up on itself, right? So then you can actually do some separation by shape. So it really depends on the separation conditions, on what sort of peak profile you would get. So actually, yes, you can separate by shape. I suppose the one thing to bear in mind with CEs as a separation technique is that if you get, for example, base replacement, so you get a change in the sequence, which doesn't affect the shape, this is something that CE can't pick up. Okay, because it won't change the length and it hasn't changed the shape, so you can't detect it by CE. So that's one thing CE can't pick up, but CE can pick up, and say clipping, where you lose oligonucleotides at either ends, the 3' or 5, the 5' or 3', for example, or it would pick up shape changes as well when you use in native conditions.
Okay, thank you, Steve. Looks like we have a few more questions. What is the lowest sample volume that has been used by CE SELEX?
Well, CE SELEX, I suppose, because it's sort of about, it's twofold here. You can talk about sampling, but you can also talk about the volume you inject. Typically with CE, you just inject nanoliters a sample, and you can inject that from a nanovial. In CIEF, we produce nanovials, which allows you to inject from as low as 5 microliters a sample. So that's typically the volume you need, but also you can collect in those as well. Okay, so you can actually collect in 5 microliters as well by having the nanovials in the collection part of the CE. So typically you can collect and inject from as little as 5 microliters a sample.
What is the lowest concentration that has been used by CE SELEX?
Typically, I think in the application, and again, you can find that on our website, is 1 picomolar is the lowest concentration we've used. So basically how it works, right, is you incubate effectively your aptamer library with your target analyte. It could be a protein, for example. And basically what happens is that the RNA molecules, which will, the target aptamers will bind with the target protein, and then the migration shifts. And typically, you can go as low as 1 picomolar of that target, say protein, for example, which is used to bind the aptamers.
And we've got a new question from the audience. Can ASOs be analyzed by CESI or CEMS?
Really good question. I think that is possible. We just simply haven't tried it yet. I think we do have plans to have projects in this space. We simply haven't tried it yet. The standard approach that we do with CEMS is sort of a CZE type separation, so it's a charge-based separation. There are other ways you can do CEMS. You can actually do it with like micelle chromatography as well. The classical CE approach that we currently use to separate RNA or oligonucleotides is via gel sieving. Of course, that's not amenable to mass specs. You don't want to push gel into a mass spec. So we're gonna have to develop a CZE or maybe a micelle approach to do the separation. Again, we're looking into this. We just simply haven't started the project yet.
Okay, looks like those are all the questions we have for today. Thank you so much, Steve. And do you have any final comments for our audience?
I think, to be honest, I think this is an area I believe is growing. I suppose, you know, if you look at some of the COVID type vaccines which are being developed at the moment, these actually are some of these are RNA based. So I think that, you know, CE will have a place in the future in this area for offering analytical techniques. And I see it also, you know, as these RNA therapy production and research and development increase, I think CE is probably going to be used more in this space for helping the analytics to test and qualify the RNA molecules. And the beauty with, I think, with CE is that it actually expands the sizes. And also we're now seeing LCMS being involved in this as well. And you know, there's probably some new workflows coming out, which will mean that LCMS will start to be used in even helping to understand the stability of the larger RNA molecules. But these will probably combine, you know, classical LCMS oligonucleotide approaches with molecular biology in order to get those answers. But I think I would suggest watch this space because I think there'll be more solutions coming out in this space, not only by CE but also by LCMS in the next year or two.
Well, thank you again, Dr. Steve Locke, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, cIEF, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.